Today I've come to talk about our nation's efforts to address this vital issue to the health and the safety of all Americans. I'm here to discuss our strategy to prevent and protect the American people from possible outbreak.
Spanish flu killed almost 1/3 of the Americans in 1918. By keeping this in mind, George W. Bush spoke in the meeting arranged by the National Environment Institution in 2005, stating, "There is no viral infection in America right now, but it may happen any time."
First, we must detect outbreaks that occur anywhere in the world. Second, we must protect the American people by stockpiling vaccines and antiviral drugs and improve our ability to rapidly produce new vaccines against a pandemic strain. A pandemic is unlike other natural disasters. Outbreaks can happen simultaneously in hundreds or even thousands of locations at the same time. And unlike storms or floods, which strike in an instant and then recede, a pandemic can continue spreading destruction in repeated waves that can last for a year or more. And one day, many lives could be needlessly lost because we failed to act today.
We should take the necessary steps beforehand instead of waiting for the virus to attack us. Though it had become the headlines in all media around the world, it was forgotten by all within a few weeks. If the presidents of the so-called developed countries had done some necessary steps to prevent the virus, there wouldn't have been many nations certifying now. When researchers advised that keeping a 2-meter distance from others will keep us from getting infected, some new updates are really scary. By analyzing their air samples from the COVID-19 patient's admission ward, it is proven that the corona can travel 4 meters, that is 13 feet from the infected person. Our generation has faced this massive virus attack, and our upcoming generation will take the virus-related research and inventions further. The reason behind that is the kind of effect the virus had caused and still causing. In a research article published in Daily Mail newspaper, researchers believe that COVID-19 is completely different from other viruses. They also believe that unlike other viruses, it changed its structure according to different human population and their immune system to spread, similar to how a chameleon gets camouflaged with its background. The article stated that the virus spreads with 3 different characteristics, namely type A, type B, and type C. Type A is very close to the virus that were found in bats and pangolins. It was the primary reason for the infection in China's Wuhan and America. This type is majorly seen in Australia and America. Type B virus was majorly seen in Switzerland, Germany, France, Belgium, and the Netherlands. Type C is different. It comes from the type B virus and it reached Europe through Singapore. We do not have an answer to the question of whether this vigorous virus is spread by human or nature. The question is supposed to be left with scientists and researchers to come up with an answer. Doctors and researchers are working day and night to save people from the virus. The researchers and medical scientists of Sankalp, the beautiful world, shared their views and comments on the same. There are 2 types of tests available to identify the virus. One is a real-time PCR assay. By collecting mucus samples in swabs from a person's nose or throat, they will test for the presence of corona gene. Coronavirus has RNA genes. Since the sample we collect has less number of RNAs, it is really a difficult task to separate the RNA from given samples. Hence, reverse transcription is being used. At first, the gene from a virus is changed into DNA. Then, the DNA is replicated into 10 of 1,000 copies. The RNA gene in the cluster is then counted. ICMR has approved only a few hundred labs in India, and also polymerase chain reaction is shortly called PCR.
According to World Health Organization, more than 70 vaccine candidates has been identified and they are under different stages of research and development. Each of these vaccine will contain an antigen either in the form of the virus or in the form of a viral antigen or in the form of a nucleic acid sequence that can able to encode that antigenic protein. Once the vaccine was introduced into a system or a human body, this antigen will elicit the immune response, will trigger the immune response, and antibody will be produced against that antigen, neutralizing that antigen and producing the immunization. There are three types of vaccines, and vaccines for SARS-CoV-2 can able to fit into any one of these categories. The first type of vaccine is using the whole virus itself as a vaccine. In this, there are two different forms. One is either using the live attenuated vaccine or using the inactivated vaccine. In the live attenuated vaccine, the whole virus is taken and the virulence is attenuated. In other words, the disease causing factors from the virus is completely removed, and then the virus can be given as a vaccine to induce immunization. The problem associated with this kind of vaccine for now is, till now we haven't identified all the factors in the SARS-CoV-2 that can able to cause the disease. So with the minimum knowledge that we have for now, if we are just going to remove the factors that we already know, there might be other factors, unidentified factors by us, which may lead to some other diseases. The second form of whole viral vaccine is inactivated vaccine, where the SARS-CoV-2 can be completely inactivated and this can be used as a vaccine candidate. Already China has begun few human clinical trials using such kind of vaccine, where they inactivate the SARS-CoV-2 and then they administer into humans in a clinical trial, where antibody will be produced against this antigen. But again, the problem associated with this kind of vaccine preparation is, we need to multiply or we need to grow the SARS-CoV-2 virus in large number of amount, large quantity, huge quantity, then we need to inactivate it. And culturing the virus in such a huge quantity may be a risky. So if you see the second type of vaccine, that is a whole protein or a subunit vaccine, a protein in the surface of the virus can be taken and that can be used as a vaccine candidate. For an example, here in SARS-CoV-2, the spike protein which is present in the surface of the SARS-CoV-2 can able to act as an antigen to prepare a vaccine candidate. University of Pittsburgh has recently developed a fingertip-sized patch containing an array of microneedles loaded with this spike protein antigen. When this finger-sized tip patch is kept on the skin and a gentle pressure is applied, the spike protein antigen loaded in these microneedles was injected into the skin as an antigen source, and the antibody is produced inside the body, leading to immunization. The third type of vaccine is known as a nucleic acid vaccine, where a gene sequence, either an mRNA or a DNA sequence that encodes the sequence for the production of the spike protein, can able to use as a vaccine. Recently, National Institute of Health along with a biotech company has tested this type of vaccine in humans, where they encapsulate the mRNA coding for the spike protein antigen encapsulated in the lipid nanoparticle to prevent the degradation of the protein, degradation of the mRNA while it's introduced into the human system. Once this gene sequence has been introduced in the human system through this way, the sequence that entered into the human cell, utilizing the biological process of the cell itself, it can able to produce the antigen inside the human system. Once the antigen is produced inside the body, it will be exposed to the immune cells to prepare or to produce the antibody against that antigen. So that's how the immunization is carried out by these type of vaccines. Though there are number of vaccine candidates identified, it may at least take 2 years. Even if you consider we are fast tracking the research, it will take at least 2 years to complete all research and developmental process and to come into action for the human use. And it may take further several years to reach the humans. This is because the vaccine has to be done in a mass production and it has to reach at least 7 billion people over the world. So currently, the best preventive medicine that we have in our hand to avoid COVID-19 and the spread of SARS-CoV-2 virus is to maintain the social distancing.
The second method is the serology test, though which we can identify whether an affected person has developed the antibody. This is an indirect test and it's done using a person's blood sample. Yet, there is a complexity in this test. It is not possible for the antibody to develop in a body with less immunity, elderly people, children, AIDS-affected person, and cancer patients. Hence, it is impossible to identify the presence of virus by testing a large population. Corona testing is not an easy task. The amount of time, money, and human power consumed by it is huge.
I'm going to talk about the serology test for COVID-19. Serology tests look for antibodies in blood. Antibodies are protein which is secreted by our immune cells that can fight off the infection. The basic principle of the test is a strip coated with a synthetic antigen. When add blood to the strip, the color line will develop if antibody present in the blood, which indicate that patient has past or recent infection of COVID-19. To develop antibodies in our body, it will take 2 to 3 weeks after infection. Therefore, this test may not be useful for diagnosis for early COVID-19 infection. The main advantage of this serology test is to deep understanding about COVID-19 infection among population, and we will get how many people have been infected with COVID-19. We can also track the people with asymptomatic and cured from the disease. The negative serology test means that person was not infected, maybe early stage of infection, at the time of antibodies may not be present in the blood, or the patient immune system may be compromised, is not able to produce enough antibodies against the infection. If the serology test is positive, means the person is a positive infection or current infection. The PCR test will further confirm that person having current infection or not. Serology test is positive and not having the current infection, then we may get a plasma from this person for plasma therapy.
In the current scenario, hydrochloroquine has been recommended for COVID-19 patients. Without enough time, it has become a huge challenge for us as the virus is spreading everywhere drastically day by day. Instead of discovering a new medicine, we must test the existing medicine against this virus. Medical scientists consider this to be the smart way. Hence, they are experimenting other medicines against coronavirus. According to a research article published by the Science magazine, Remdesivir is showing some positive symptoms against COVID-19. It stops the virus from doubling. From now, the American pharma company Gilead Sciences is manufacturing Remdesivir, and unfortunately, India don't have one.
In the interest of time, repurposing of drugs that are already approved for use is considered to be the smart strategy to come up with the treatment option for COVID-19 rather than identifying a new drug molecule and evaluating it right from the scratch. Of all the treatment options being evaluated worldwide by several clinical trials, most of them fall under the repurposed drugs category. So to list a few of them, Remdesivir, it is an antiviral agent used against Ebola, SARS-1 and MERS virus. So when this Remdesivir was administered to COVID-19 patients on a compassionate basis, the results were potentially beneficial to the patients. So we need more evidence-based, conclusive findings from structured clinical trials. So the next one is the lopinavir-ritonavir combination. It is a protease inhibitor drug used to treat HIV infection. So far the results from clinical trials have been ambiguous. So we have to wait for more ongoing clinical trials to come up with the results. So next, importantly, chloroquine and hydroxychloroquine. So these drugs have received so much of media attention recently. So this is based on promising results from several in vitro studies and a few clinical trials. So this is not a new drug. They are around for more than 70 years. They are used to treat malaria and other autoimmune disorders. Recently, a study from France had evaluated a combination of hydroxychloroquine and azithromycin, and the results have been so encouraging. So based on this outcome of French study, several treatment guidelines have been formulated. Even the Indian Council of Medical Research had recommended the prophylactic use of hydroxychloroquine for healthcare workers who are treating COVID-19 and for the close contacts of laboratory confirmed COVID-19 patients. But one thing is to be noted is that these drugs, hydroxychloroquine, chloroquine and azithromycin, they have several side effects and toxicities. So the patients who are prescribed with this have to be monitored closely, especially for their cardiac health. So the World Health Organization has currently launched an international clinical trial known as Solidarity Trial. So this includes 4 treatment options. It evaluates 4 treatment options. Most of them are the repurposed drugs. So these treatment options are evaluated against standard of care. So what is so special about this clinical trial is that it is partnered by 100 countries and it enrolls like more than thousands of patients. So the advantage is that it can save a lot of time, and the whole world is awaiting the results of this clinical trial, and hopefully we can get a very efficient treatment option against COVID-19.
We all have this question that when corona belongs to the SARS family, why the medicine given for SARS is not beneficiary?
SARS and COVID-19 are 80% similar in their genetic material. You may ask if the developed SARS vaccine can be used against COVID-19. The answer is no. It is because the developed vaccine is an inactive virus, meaning that it cannot cause the disease. When this SARS virus is given as a vaccine, the individual produces antibodies. So when this same individual happens to be exposed to SARS again, the preformed antibodies, they attach to this virus and block them from entering the body cells. If you see where these antibodies attach, they sit precisely on the spike-like structures called crowns on the virus. All coronaviruses differ in this crown region, and hence the SARS vaccine cannot be effective for COVID-19.
The reason for the great anxiety is the fact that corona spreads many times faster than SARS and MERS. When the infected person sneezes, there will be water droplets that spreads quickly. Also, touching a place or object used by an infected person is responsible for rapid spread. Hence, the medical world has jumped into action to develop medicine for COVID-19. When there is pandemic, various kinds of research are made. Especially, stem cell research is most important of them all, suggests scientists.
Stem cells are unique cells that have remarkable potential to differentiate into many different cell types in the body. If you see, there are different sources of stem cells. They are bone marrow, umbilical cord tissue and adipose tissue, that is fat cells. If you see, after the year 2000, the world has faced the outbreak of viruses such as MERS and SARS. However, the clinical studies on the use of stem cells for virus-induced organ damage has not been properly addressed. If you see, after the outbreak of coronavirus in December 2019, a collaborative study has been done in China, and the reports of this study has been published in the journal Aging and Disease. This is a pilot study which consists of 7 patients. Among the 7 patients, 1 is critically severe with coronavirus infection like pneumonia, and 2 had mild to moderate symptoms of coronavirus infection, and 4 had severe symptoms. Upon the injection of medical grade mesenchymal stem cells, these patients showed the improvement in their pulmonary function and the increase in the anti-inflammatory cytokines such as IL-10 and decreased in the pro-inflammatory cytokines such as TNF-alpha, and also the CRP levels were decreased. After the 10 to 14 days of period, these patients were discharged from the hospital. If you see, as a stem cell researcher, we feel that this is a pilot study and large population studies has to be done in order to justify the use of stem cells for coronavirus infection. And other limitations of the study are the mechanism of mesenchymal stem cells has not been explained, and also the data related to the effective immune response and modulation has not been explained. After this outbreak of coronavirus, around 30 to 35 clinical trials has been initiated and 2 studies has been approved by the FDA for the clinical trial. Among that, one study has been done in the Miami state which involved around 24 patients, and these 24 patients are coronavirus infected patients. For them, the umbilical cord stem cells will be given and they will see how these umbilical cord stem cells are improving the symptoms of this coronavirus infection. Likewise, another study has been done in the New Jersey biotech company. This study involved the natural killer cells. That is, the placenta was the source for these natural killer cells. This is a pre-stage trial where the patients are having a mild symptoms of coronavirus. The purpose of this study is to inject the natural killer cells and to see whether the injection of these natural killer cells will improve the symptoms of these patients and prevent them from going into the severe illness. However, the scientist in the field of immunology has warned this kind of studies, the injection of natural killer cells, because these natural killer cells sometimes may activate the hyperimmune reaction which will worsen the existing respiratory problems.
Pharmaceutical companies such as CureVac, GlaxoSmithKline, Inovio, Regeneron, Johnson & Johnson, Vir Biotechnology are now in the efforts to prepare medicine and vaccine against COVID-19. America-based private pharmaceutical company Arcturus Therapeutics had told that they have successfully developed the vaccine for COVID-19. Yet, it is said that they are waiting for the vaccine to be tested and licensed, which requires some time. It is not an easy task to market the medicines since it has to pass hurdles. Countries like China and America are at the stage of experimenting the vaccines in humans.
It's been more than 4 months since COVID-19 have been discovered. You may wonder why there is no vaccine yet. It takes 5 to 7 years to make a new vaccine. There are several stages during this process and each stage has a specific purpose. First, a vaccine candidate is tested in the laboratory on cells, followed by animal testing. Then it enters into human clinical trials, which itself has 4 different phases. During all these various stages, a vaccine candidate is tested for dosage, safety, side effects, and efficacy. Currently, there are more than 60 vaccine candidates in preclinical testing and only 2 have entered into phase 1 clinical trials. The first one was made in China and is an adenovirus-based DNA vaccine. The second one was made in USA and is an RNA-based vaccine. It is an interesting approach since RNA vaccine have now been approved.
The major complex situation here is that most of the people who are affected by coronavirus show not much symptoms. This makes many vulnerable to the infection without any knowledge of getting infected. Often, they function as carriers spreading the virus to the fellow beings. Scientists express their concerns that severe acute respiratory syndrome coronavirus 2, also called as SARS-CoV-2, will be a huge challenge to human beings.
It will be a great challenge to manage the COVID-19 disease even after the pandemic situation and new vaccines development. The reason being, first, the vaccines which we may get from the other countries may not work efficiently for us. Since this RNA virus has been shown to get frequent mutations in their genomes. Moreover, we do see such mutations in our isolates from India, suggesting that we should have one specific to our populations. The second reason is the protective immunity of the people who gets infected but doesn't show any severe symptoms and may not stay for a long. Moreover, the recent data shows that resurgence of the infection is unpredictable. So how do we solve these great challenges? First, most importantly, identify the groups of patients who respond to the disease and who is not. So how do we do that? with all kinds of data such as clinical image data, for example, lung X-ray, medical records, biochemical parameters and other parameters. We can stratify the patients into no, mild, moderate or severe disease. Even further, the severe disease groups can be further categorized into multiple groups such for example, patients having respiratory problems with no lung damage, and patients having multiple organs which gets infected through a spread of COVID-19 disease. For example, the recent research shows that tissues which has cobalt or ciliated cells as an entry point for the virus. So in this case, they are high also as the cobalt cells has a possibility to get infection. And the clinical data as mentioned show that 1% of the general population who gets infected is expected to have IE infections. The next most important, we are talking about the comorbidity. In this case, for example, 1% of the cancer patients of the general population has been shown or prevalent for this disease. If that is the case, we can even stratify the cancer patients who gets severe infection, who is not. If you do that, this will help to identify new biomarkers for early diagnosis and prognosis of the disease through comprehensive research involving basic scientists, clinicians and bioinformaticians. This would ultimately help to manage the disease further and also help to identify new therapeutic targets.
In India, the number of people getting infected increases day by day. Many medical researchers state that there is a need to test more people. They also add that there are a lot of assumptions about the disease in India which are not scientifically proven.
Through this video, I would like to share some information on COVID-19. The first one is why the number of cases are reported less in India when compared to other countries globally. Out of many reasons, in which important one is the testing method. Until last week, we are confirming this infection virus through the procedure called Rt-Pcr method. This test involves various steps and it takes 5 to 6 hours time. And we are doing this test only for the patients who is having symptoms of corona. And we are doing this test for very less number of patients because of the tedious procedure. Now, Indian Council of Medical Research has approved a test called rapid test, which is very simple test which is confirming the COVID-19 qualitatively. To do this test, it will take only 30 minutes. By using this test, we can test the infected people and also their surroundings. And the next one is high temperature. Usually in India, there is a talk among the people that the coronavirus will not withstand in high temperature. That too, India is in the early stage of summer, so the coronavirus will not virulent in India. But actually, the Middle Eastern countries like Dubai, Saudi Arabia, and other are reporting more number of cases every day. So this high temperature will not. This hypothesis is not proved scientifically. The next one is BCG vaccines. The BCG vaccine is a compulsory vaccine in India. Most of the Indians have taken this vaccine at their childhood days. So there is a say among the people, this vaccine, those who have taken this vaccine, has the capacity naturally to prevent the coronavirus infection. But this also not proved scientifically. And the next one is, nowadays the Indian people are talking about a drug called HCQ, that is hydroxychloroquine, which is used for malarial infection. And many Indians have taken this malarial infection. They taken this drug for malarial infections, and the people are telling this drug will prevent the coronavirus infection. And this also it's not proved scientifically. And next is about the coronavirus. The coronavirus will naturally undergo many changes genetically. This is called mutation in science. This virus sample taken in Wuhan, China, and the virus taken in India and submitted in NCBI have few differences. See, for example, in the virus sequence, in the DNA and the RNA, at the 930th position, amino acid glycine is replaced by valine. This may be the reason for less virulence of virus in India, and this may be the reason for the asymptomatic patients in India.
Many countries are trying to provide plasma therapy by separating the antibodies from the plasma of the patient's blood who recovered from COVID-19. The purpose of this is to cure an infected person by using the antibodies from the recovered person. America, China, and South Korea are already into it, and in India, Kerala is the only state that have planned to experiment this first. Yet, there are some limitations too, such as these antibodies are absent in those who are asymptomatic. Studies suggest that either one should have the virus for 21 days or severely suffered from the virus to develop the antibody. We can collect plasma only from these people after recovery. If those plasma proteins are injected into an infected body, there are much more possibilities for them getting cured. Medical researchers and doctors say that they can come to a conclusion about plasma therapy's effectiveness only by doing a large population clinical trials.
Here we are going to talk about corona and cancer. First, as we are going to discuss about the corona, it is first reported in China in December 2019. Has mutated into 10 different types, and one of them is A2A, has nearly replaced all the other to become a dominant across all geographical regions. The novel coronavirus with the A2A mutation is highly efficient in entering human lung cells in large number. And this previous SARS has killed around 800 people, but now at the current COVID-19 has killed more than 50,000 people. The major entry path is to the lung, and it infects the receptor called as a ACE2 receptor, that is an angiotensin converting enzyme 2, which present in the lung is a major receptor for binding of this virus to the lung. And A2A enters lung very efficiently. And the global spread, if you look, it's like 50.8% is around the A2A mutation. And there are around 10 types. From the ancestry type is the O. And there are different types as O, A2, A2A, A3, B, B1, and so on. The global pandemic of coronavirus disease called COVID-19 is having a serious impact on many people, including cancer patients, their family, and caregivers. Cancer patients are among those at high risk of serious illness for an infection because their immune system are often weakened by cancer and its treatment. Most people who were treated for cancer in the past, especially years ago, are likely to have a normal immune function, but each people is different. It is important that all cancer patients and survivors who are currently involved in the treatment need to talk with their doctors to understand the situations and their medical histories, because each and every person is unique. It is also important that both patients and their caretakers precaution to lower the risk of COVID-19. While the news about outbreak is changing every day, knowing some basic facts about what can and cannot be done to help protect you and your from getting sick can be very empowering. How can I protect myself and others from getting the COVID-19? As we know, all the precautionary measures, as the health officials is mentioning, avoid touching the eyes, avoid close contact, avoid shaking hands, staying home when you're sick, clean and disinfect frequently, avoid non-essential travel. Cancer patients need to know about the coronavirus. The COVID-19 outbreak is still fairly new, so doctors do not have a lot of specific information on this coronavirus for cancer patients. But if they do have a lot of information regarding the risk of infection in general for cancer patients, avoid being exposed to this virus is especially important for cancer patients who might be at higher risk for serious illness if they get infected. This is particularly true for patients who are getting chemotherapy or stem cell bone marrow transplant because their immune system can severely weaken by the treatment. The CDC, that is Center for Disease Control, is recommending that healthcare facilities and doctors and dentists urgent emergency visits and procedures for the several weeks are under strict regulations. Are there medicine to treat COVID-19? Yes, at this time there are no FDA-approved drugs to treat COVID-19. Although some medications might be helpful in treating symptoms from the disease. The drug Chloroquine and Hydroxychloroquine, which are already used to treat malaria and other conditions, are being studied for the possible mechanism to treat the COVID-19. Because they are already available to treat other diseases, some doctors are trying them in certain patients with COVID-19. These medications can sometimes cause serious side effects such as heart rhythm problems, so they should be taken only under the close supervision of doctors. In fact, FDA is cautioning against the use of this drug for COVID-19 unless a person in the hospital or is taking particularly in clinical trials. Several drugs that might help treat COVID-19 are also being studied in clinical trials, but they are under the clinical trials, and vaccine trials are undergoing for the treatment of the new COVID-19. But every day, it's the bottom line, scientists are learning more about the virus every day, and health experts are updating their information daily. What are the symptoms of COVID-19? The most common symptoms of COVID-19 is which may appear 12 to 14 days after being infected with the virus. Fever of at least 100.4 degree Fahrenheit, that's 38 degree centigrade, cough, shortness of breath, chills, shaking with chills, muscle ache and pains, sore throat, headache, new loss of smell or taste. Other symptoms can include feeling very tired, loss of appetite, and coughing up sputum. The latest corona guidance for people living with cancer. The most important thing is to follow the advice from your healthcare systems. People with cancer may have a high risk of getting COVID-19. So the advice, now they are just usually following 3 S's. One is social distancing, one is shielding, and one is self-isolation. Social distancing, as you know, is like distance which you're maintaining from one person to another, or you can call it as a physical distancing. And the shielding is usually given by the government guidance, where the people at very high risk of illness for the coronavirus because of their underlying health conditions. Self-isolation, if you live in the household with a possible corona infection, you need to self-isolate yourself. The other primary question we have, are people with cancer more vulnerable to coronavirus? Some people with cancer and those who have received or receiving certain treatments are more risk becoming seriously ill with the COVID-19 infection. People undergoing active chemotherapy, people having immunotherapy or other continuing antibody treatments for cancer, people having other targeted cancer treatments which can affect the immune system such as protein kinase inhibitors or PARP inhibitors, people having intense radiotherapy for lung cancer, people who have bone marrow or stem cell transplant in last 6 months who are still taking immunosuppressive drug, people with cancer of the blood or bone marrow such as leukemia, lymphoma or myeloma who are at any stage of the treatments. The other question is like, cancer affects my immune system? If the people have the cancer, the immune system in the body defends against bacteria, viruses and other foreign organisms and harmful chemicals. The blood and the lymphatic system are part of the immune system. Some type of cancer can affect the immune system, for example, blood cancer such as lymphoma and leukemia. Some cancer treatments affect the immune system. These include chemotherapy and immunotherapy, radiotherapy may sometimes affect the immune system. Most people's immune systems will recover well. They are finished with the treatments. Next, what will happen to my cancer treatment if you ask yourself? If you're having a cancer treatment now or preparing to have it soon, it is understandable. If it's worried about how coronavirus might affect you, your clinical team is the best place to talk with about the effect, about the treatments, and get a possible appointments if you have a severe risk and you are affected by the COVID and you have early symptoms of COVID.
Only the doctors, medical researchers and the scientists are required in large numbers. All governments in the world are looking at them for help because only they can restore the world for us from this pandemic. Let's hope for the best.